This class. Okay, so also to tell you, Hafsa, we are doing genetics at the moment, and today we'll be starting the 6th chapter, which is molecular basis of genetics. And after the class, can you just stay back for 5 more minutes so that I can discuss with you on the curriculum and the plan ahead, Hafsa, just for Hafsa. Okay.
Yes, sir, sure.
Cool. Okay, how are you, Parker?
I'm good, sir. I'm really sorry I joined late.
No, it's okay. You just joined in time. Okay. But if you feel really guilty of joining late, how about you compensate by giving a quick summary? There's always a treat for all of us in that. Just a quick summary of what we talked about in the last class. I think we talked about pedigree, and I gave you some homework, right? So would you like to just orient all of us to that? I'll be there with you if you miss something, no worries.
Yes, sir.
Okay, great.
So we studied about pedigree analysis last class, and we did some sums. We learned how to find the allele and find if it's dominant or recessive, and we learned how to find if it's X-linked or autosomal.
Right, and I gave you some. No, please finish. Go ahead.
We also learned how only females can be carriers and males cannot be carriers. Males can either be normal males or affected males. And
but for what? All these statements are context dependent. You are right about all these statements are correct. But I told you that males can never be carriers for a particular type of inheritance, not for all types of inheritance that we study through pedigree. So in what kind of inheritance males cannot be a carrier? And what is the reason behind it?
X-linked.
I think we discussed it here, right? Yeah. Yeah, go ahead.
Yes, sir.
Yeah, so you said X-linked, right?
When it is X-linked, yes, sir.
Yeah. Yeah, go ahead, go ahead. Sorry, I think there is a lag, so that's why.
When it is X-linked, then both the X is supposed to be affected for the female to be affected, but if only one X is defected, then she's a carrier.
Correct. But in case of dominant X-linked dominant traits, even a female can be affected by just having one affected X, right? One mutated X.
Yes, sir.
Yeah, right. Yeah, and then what we discussed? Remember, I gave you some.
We discussed about.
I gave you some shorthand tips, like when we are looking for a dominant pedigree, what to look for. When we are looking for a recessive pedigree, what should we look for? When we are looking for autosomal diseases versus sex-linked diseases, what should we look for?
It's when F1 skips.
Go ahead. Go ahead.
One generation is skipped when it is.
When it is
dominant.
Recessive. If it is dominant, it will dominate in every generation, right? For anything to be dominant, it just needs one allele that is mutated or that is changed, and the character will be shown, the phenotype will be shown, even by just having one allele. It is exactly similar to what we discussed before in the chapter. When Mendel figured out something was capital T, small t, this genotype will always give rise to tall plant, right? This allele will not be
Yes, sir.
expressed, right? So that's what dominant means. So dominant does not skip generations, but recessive traits skip generations. So in this case, since you can see all the generations have the disease in some or the other individuals, one thing which is very clear is that it is a dominant trait. Whatever it is, it's a dominant trait because all the generations are showing the phenotype, correct? Versus I gave you another
Yes, sir.
pedigree here, and we figured out that it is sex-linked and recessive. But one thing which is very obvious is this generation is skipped. Okay, there is electricity is lost, but just bear with me. So this generation is skipped, and you can see that the phenotype again shows up in generation 2, 3, and also in 4. But in 1, there is no phenotype. So it's a recessive disorder to begin with. Make sense?
Yes, sir.
Yeah. And yes, continue.
And if it is autosomal,
Mhm.
the defective the disease goes from sons to mother. Also, sorry, sons and daughters. And if it is uh, X-linked, that then it only goes to the sons
From?
from the mothers.
No, that's not true. It's it can also go to daughters, given that let's look here. So I understood what you are saying. You're not just framing it in right words. So I'll just repeat it once more for all of you, and please keep taking notes whenever you feel like. Okay. So see, when we say sex-linked diseases, there are two sex chromosomes, X and Y. But in your syllabus, we will only deal with disorders which are associated with X chromosome, not with the Y chromosome. There are reasons for it. Y chromosome is the smallest. It has the least genes, and almost all of the genes on Y chromosome are associated with the male characters, the male secondary sexual character, or regulation of the secondary sexual and primary sexual characters. So that's why we are not studying diseases associated with Y chromosome. But let's do a exercise. Let's say I am talking about Y chromosome disease. Sorry. In which case, just 1 second. Yeah. If a male has a defected Y, it will look like this. But female, since females have no Y, they will be X and X, right? And if these two are mated together, then it is very obvious that this male, who's the father here of generation 1, can only give this disorder or this disease or this phenotype to sons, right? Because male gives Y chromosome only to the son. Females do not have a Y chromosome. So it is the easiest to figure out a pedigree of a Y-linked disorder. You will see father having it, and then the son, and then the next son, and then the next son. It goes from, it goes in all the males of the lineage. So no point in studying it per se. But if it is X chromosome, so males just have one X chromosome, right? So it will look something like this. It will have a mutated X. I'm using the red color because red color signifies some kind of a problem or danger. So X is problematic here. But if in the female also there is one X which is problematic or mutated and one X which is normal, so this male, will it be a carrier or or a affected
No,
individual?
affected individual.
Yes. So males can never be carriers for a X-linked disorder, okay, or any sex-linked. Even for a Y-linked, they cannot be a carrier because they just have one Y chromosome and one X chromosome. So if this X is mutated, nothing can rescue the phenotype, and whatever gene is affected, the effect will be shown, or it will just appear in the phenotype. So this is an affected male. And let's say this disease is X-linked, but it is dominant. So if it is a dominant disease, male will be also affected and this female will be also affected, right? Make sense?
Yes, sir.
Yeah. So, and if this female gives this X chromosome to son, that son will be also affected. And if this female gives this affected X to a daughter, daughter will also be affected. So it's not true that X-linked disorders can only pass from mothers to son, right? In case of X-linked dominant. But let's say it is X-linked recessive. So in case of X-linked and recessive disorder, what does recessive disorder means? Again, quickly.
Both
Parker?
the recessive skips one generation.
Yeah, but for someone to have that disorder, what the person needs to have?
Both their alleles should be defective.
Exactly. It needs 2 copies of the defected allele, right? For the affected allele. In this case, 2 copies can only come from to a daughter from both, 1 copy from the father and 1 copy from the mother. So in the progeny, if they have 1 daughter who got the defected copy from father and defected copy from mother, this daughter is going to be affected, right?
Yes, sir.
Let's say there is another daughter who got 1 affected copy from father, but she got normal copy from the mother. Will she be affected or carrier? It's X-linked recessive.
She will be a carrier.
She will be a carrier. And let's say they have 1 son as well. Now son gets X chromosome from mother and Y chromosome from father. Now mother has a defected X. So if a son gets a defected X, the son will be?
Affected individual.
Yeah, affected. But if the son gets a normal X from mother, then?
He's a normal individual.
Right, normal individual, correct?
Yes, sir.
Yeah, this is a carrier and this is affected. So do you understand it? We have to trace back what X the son is getting from the mother. Okay. Based on that, it will be decided whether the phenotype will be transferred to the son or not. Yeah. Is it clear?
Yes, sir.
Everyone? Afsa, Mariam?
Okay.
Yeah. Okay, perfect. So just this was just the recap. So and I gave you homework to go and read the chromosomal disorders, Mendelian disorders. Did you all go and did you all go back and read it? Yes, no? Okay, so let's start with a few that you have studied. So first I'm talking about Mendelian disorders, which means they are associated with genes and not chromosomes. So we also call them genetic disorders associated with genes. Okay, so one that we already talked about, remember, which was phenylketonuria. Anyone remembers what did we discuss about phenylketonuria? Yes? No? Okay, so what kind of disease, what kind of disorder is phenylketonuria? Sex-linked, autosomal, recessive, dominant? I think you read this part, right? I gave you as a homework, people. I can give a free pass to Hafsat for today because she just joined in, but others, I'm expecting answers. Come on. It is autosomal, sex-linked, what it is. Okay, let's talk. Let's see it like this. So yes, Iram says it's autosomal. Correct. It is an autosomal disorder. Sex dominant or recessive?
Recessive.
Yes, it is recessive, and it's good that it's recessive. Okay, if it was a dominant disease, then people would have, like humans would have just died very early on, even with just one defected copy. Okay. Now, what happens in phenylketonuria? If you remember, I told you there is amino acid in our body called phenylalanine, right? Amino acids are building blocks of proteins, and we get these amino acids through diet, through the proteins that we eat. So we first eat protein, we break it down into amino acids, then we absorb all these amino acids and remake proteins that we need in the body. So phenylalanine is one such amino acid, and this amino acid is converted into tyrosine, right? Remember I told you this? And to do this reaction, we need an enzyme which is called phenylalanine hydroxylase. This is the enzyme that is responsible to drive this reaction. Enzymes are just like biocatalysts, and there is a gene in our body which codes. So the gene that codes for this enzyme, phenylalanine hydroxylase, it is called PAH. So this gene gets mutated. Okay. So because of mutation in the gene that forms this enzyme, now phenylalanine cannot be converted into tyrosine. So what will happen? The levels of phenylalanine will start rising up. And then this phenylalanine, in the absence of this enzyme, will convert into phenyl pyruvic acid in the body. And this is problematic. Okay. Do you understand that, everyone? This is not good.
Sir,
Sorry. Yes, go ahead.
can you please explain again?
Yes, I will. So we have, I thought we did it before, so you people might be remembering, but no problem. Okay, we can go over it as many times as you want. Just remember, so it is phenylketonuria. First, let's go through the name. The word phenyl comes from phenylalanine, okay, which is amino acid. Ketone, you know the ketone bodies, right? Ketone groups. Have you studied functional groups in your chemistry class 11th, I guess?
Yes, sir.
Carboxylic
Yes, sir.
acid group, aldehyde, alcohol. So you must have studied a ketone group also, right?
Yes, sir.
Double bond O.
COH.
Yes, double bond O and one H. And if it is COOH, it is carboxylic acid, right?
Acid.
Yeah. So ketone bodies are very reactive. They're not good. It should not stay as ketone bodies in our system. So in this disease, what happens is you find ketone bodies, the high level of ketone bodies in urine. So the name phenylketonuria tells you about then in this condition, the enzyme phenylalanine hydroxylase is faulty, because of which the normal function is not happening in the body. The normal function is this enzyme helps phenylalanine to be converted into tyrosine. Tyrosine is another amino acid. So this is also amino acid. This is also amino acid. And then after this conversion, you do not get very high levels of phenylalanine. And if this enzyme is not present, which is the reason for this disease, where the gene that codes for this enzyme, so genes make proteins, enzymes are basically proteins. So this gene is mutated, the gene for phenylalanine hydroxylase. Remember that gene cannot be for phenylalanine. Phenylalanine is an amino acid, but phenylalanine hydroxylase is an enzyme. How to identify enzymes? First, very easy method is they have ASE, ASE in their names at the end. Okay. This also I discussed. Remember, class 11th, all the enzymes that you have studied in the digestive system. Can you name any enzyme from digestive system unit? The chapter digestion, was it there in your syllabus? I think it was, right? Or was it removed? Did you study digestion and absorption?
So it was deleted.
Okay. That's
Sir, it was
the
not there.
beauty. It was not there, right? That's the beauty of it. So they remove chapters, but they're all connected. So that's very, it's difficult. So, but you remember, even forget about that chapter. Do you remember what's present in our saliva that helps in digestion?
Salivary amylase.
Perfect. Salivary amylase. So the enzyme's name ends with ASE, amylase. If you have to use an enzyme to digest protein, it will be called protease. Okay. If you have to digest lipids, it's called lipase. If you have to digest nucleic acid, it will be called nuclease. Okay. So yes. So enzymes end with ase, and this enzyme, what it does, it takes phenylalanine and hydroxylase. So it reduces, it adds hydroxy group to the phenylalanine and makes it into tyrosine. So this is the normal function, which after mutation in this gene does not happen. But remember, this is recessive, a disease. So you need both the mutated copies, one from father and one from mother. Then only this metabolic disorder can happen. So it is a genetic metabolic disorder, and it will happen by birth. So it's called an inborn metabolic disorder. We can write down phenylketonuria is an inborn or by birth metabolic disorder. Because what now does not happen is phenylalanine does not convert into tyrosine, and if it will not convert, its level will start rising in the body, and then body will try to do something with it, and it will end up making phenyl pyruvic acid, and it is a very, very toxic substance to accumulate in the body. And because of this acid, there are multiple things that happen. So this example we also studied in which part? Remember, if I go up, we studied phenylketonuria. Where? Here. Remember this, people? Can you see the board, everyone?
Yes, sir.
How can no one remember this? I'm sure I was teaching. There were some of you must be there. I was teaching this. It's on the board. So phenylketonuria is also an example of pleiotropic inheritance, which means a single gene can affect or regulate multiple phenotypes. So the single gene, when it gets mutated, it affects mental health. So there is mental retardation because it affects the brain. There is also a skin pigmentation because it affects skin follicles. So skin depigments or overpigments. There is also hair loss and hair pigment depigmentation because it affects hair follicle. So just one gene affected and it affects lot of phenotypes. So that's called pleiotropic inheritance. Okay. And it's also a Mendelian disorder because it happens by birth due to mutations in the gene. So it is one thing that you are studying for 2 topics, makes it better for you. So if you remember this, it's good. And in this, what happens? This phenylpyruvic acid gets accumulated after it is formed. If it gets accumulated in brain tissues, you see mental retardation. If it gets accumulated in skin cells,
So,
yes, Iram.
what's a retardation?
Uh, Retardation means slow or abnormal growth. Retard is the word for where, where anything is inhibited from growing properly. So their brain does not develop. These kids, when they are born, their brain does not develop as it should have been. So there are many centers in the brain which are underdeveloped. So these kids have difficulty in learning, memorizing, even sometimes speaking, because the speech center is not properly developed. So they cannot formulate proper sentences or words. They cannot learn if you tell them something. Hence, they do not learn how to behave or how to, behave in certain different conditions as well. Okay, or how to communicate properly. That's called mental retardation. Is that clear?
Yes, sir. Yes.
Yeah. And if it accumulates in skin cells, there is skin depigmentation. And if it accumulates in hair follicles, there is hair loss and also hair depigmentation. Okay. Apart from that, uh, one symptom of this disease is how it is identified most of the times is that you see uh, there are a lot of ketone bodies in the urine. So you find ketone bodies in high amount of ketone bodies in the urine. And because of that, kidneys cannot function properly. So kidneys have very poor absorption. So write down. Patients, the kids with phenylketonuria also have poor absorption rates of kidneys. So this is one Mendelian disorder. Okay. And the gene is located on an autosome, not on a sex chromosome, and it is a recessive disorder. Yes. What's the other one? Quickly. So I see that you people have not. Sorry. Yeah, go ahead.
Yeah.
Come again, come again. I could not understand. The voice is a little muffled.
Sickle cell anemia.
Sickle cell anemia. Okay, perfect. So let's go to sickle cell anemia. So you have to tell us what is sickle cell anemia. Again, look at the name. Sickle cell anemia, right? Anemia simply means there are many kinds of anemia. It means lack of hemoglobin or low RBC levels. So what is happening here? Why it's called sickle cell anemia?
RBCs get converted into sickle shape.
Yes, perfect, Afsa. So the shape of RBC should be a discoid, biconcave disc-like structure, but it becomes sickle-like. So its surface area reduces drastically. Okay, so a RBC that should look like this converts into something that looks like this, right? So you can see that it has flattened and become a sickle. So that's called sickle cell anemia, because of which the hemoglobin level goes really down and the person does not have healthy hemoglobin, proper hemoglobin to transport oxygen everywhere in the body. Now, what kind of disorder it is? It is another autosomal and recessive. So to cause the disease, you need both defected genes. Okay, here also. But there is a little some difference here. So what happens in sickle cell anemia is, if let's say the gene for hemoglobin, write down, um, this disease is controlled by a single pair of allele. Okay, so there is one single pair of allele and we call that allele as HB. HB is for hemoglobin. Okay, it's controlled by single pair of allele HB. So normally the normal allele is HBS, sorry, HBA. Okay, so this is a normal allele which give rise to normal hemoglobin, and the other is HBS. This is called the sickled allele or the mutated allele. Okay, and what happens here is just a very a very simple mutation, which is also known as point mutation. I talked to you about point mutations, right? So this happens due to point mutation, just one single mutation at one single position in the whole sequence of this gene. Now, in this case, the normal gene HBA has this sequence, GAG on one strand, and on other strand, what should be it? CTC, right? Because they are complementary. So this is the position. This codon GAG and CTC gets converted into, in a sickled allele, into GTG, and on the other strand you will find CAC. So from CTC it becomes CAC. And let me tell you, CTC will give rise to a mRNA. So this is DNA, okay? This is DNA. This is also DNA. From DNA, mRNA is formed. So this strand is red to form mRNA. And when mRNA will be formed, it will have GAG here. And in this case, it will have GUG, right? This is the mRNA. You don't have to remember all of it. You just have to note that GAG as a codon forms glutamic acid. And GUG forms valine. So this is the change. Just one base changes. And when you are making the protein, in that protein chain, let's say instead of glutamic acid, you have a valine. Okay. And this change leads to a disease in the a change in the whole structure of RBC, and you get a disease. So sometimes, remember when we discussed mutations, let me go back to that part. Yes. Do you remember mutations? We discussed mutations, and I also told you that point mutation is where, like, which is very, very problematic because it changes the whole protein. Okay. And sickle cell anemia is one case. Is it clear, everyone?
Yes, sir.
Okay. Now,
So,
if it is yes,
sir, so
yes, Partha.
if instead of GAG forming glutamic acid, it forms valine.
Yes. So because instead of this A here, um, you see this A here, you have a
Yes,
T
sir.
here. So if you change this A to T in the DNA, then on the other strand, the T will change to A, right? It's obvious because
Yes, sir.
complementary pairing is there. And because of this change, when you will read the DNA to make RNA, similarly, the T here will be changed to A again, and the A here will be changed to U because mRNA has a uracil, it does not have thymine, right? mRNA has AUGC, DNA has ATGC. And when you read, when ribosomes read this, so during translation, when you are making proteins, so from mRNA, protein is formed, right? DNA, RNA, protein. Remember that? And to make
Yes, sir.
protein, ribosomes are important. So when ribosomes are reading this mRNA, now there is a mistake which is not corrected. It cannot be corrected because there is a mutation in the body. And when the ribosomes will read this code, GAG, normally they would have added a glutamic acid, amino acid. Okay. But now because of the mistake, they will read GUG and they will add valine, not glutamic acid. So just by adding one different amino acid, the whole structure of the hemoglobin protein and the whole structure of the RBC changes. And from perfect discoid shape, it becomes a deflated sickle cell. It's just like you take a balloon, take one single pin and poke it. What will happen? It will
It
deflate,
will
right?
burst.
So yeah, one single change and a very huge result. So that's what this point mutation does. And so we understood that a normal person will have HBA and HBA, right? These two copies from both the parents, correct?
Yes, sir.
And we also know that a diseased person who have sickle cell anemia, what will be the genotype of a diseased person? HBS and
Yes.
HBS. Now let me ask you, what will happen if a person has one normal copy and one diseased copy? So this is a homozygous condition, right? And this also is a homozygous condition. So if the homozygous condition is of a normal allele, it will be a normal person. If the homozygous condition will be of the dominant allele, it will be a affected, of the mutated allele, it will be a affected person. That's how recessive diseases work, right? Recessive phenotypes have to be in homozygous form to show their effect, just like small t, then only it can make a dwarf plant. Remember that?
Yes, sir.
Yeah. So, but in this case, normally what should happen? Tell me. This is dominant. HBA should be dominant over HBS and the person should be normal, right? Yes or no?
Yes, sir.
Yes, sir.
Yeah. But instead we call them carrier people, like they are carrier persons. So those who have a heterozygous allele, one mutated and one normal, they are carriers. And it has been seen that almost around half of their RBCs are normal shaped and the other half is sickle shaped. Now, this is where we understand that It's not true dominancy, right? Remember incomplete dominancy? Yeah. Remember incomplete dominance? People, laws of Mendel. I hope you're not forgetting what we are studying in the past.
Yes, sir.
Yeah. And what is interesting and what comes in an exam is that there is a disease called malaria. How many of you know about malaria? Dengue, malaria. Yes, Parker, tell us what happens in malaria.
It's caused by
What causes malaria?
Mosquitoes.
Mosquito causes malaria. Okay. Mosquito also causes dengue. Mosquito also causes chikungunya. But is it mosquito really or something else?
Female and
Female Anopheles is the vector, the mosquito that carries a pathogen known as Plasmodium. Plasmodium is a protozoa, a microorganism that causes malaria, right? And you know what is the target of malarial parasite? To increase their number to replicate in our body. I'm just going a little away from the main thing to prepare you about another concept. So all the knowledge is connected. So the target cells are RBCs, red blood cells. Have you ever seen a malaria patient? Anyone who unfortunately have suffered from malaria or have seen anyone suffering from malaria in the family or friends? I'm sure we all have seen someone suffering from malaria. I myself suffered from malaria once. Anyone who has seen or known someone who suffered from malaria?
Sir, I have not seen, but I have a cousin who suffered from malaria.
Okay, so do you have any idea about symptoms? What happens in malaria? You get recurrent cyclic.
Fever?
Yes, you get fever, of course, but you also get recurrent cyclic chills, like this feeling of cold and chills and shivers in the body. And it is exactly around 42 to 36 to 42 hours that the cycle repeats. Every other alternate day, you will feel very, very shivery and chilling. And that is because that is the time that the malarial parasite takes to attack RBCs, replicate inside the RBCs, and burst the RBC to release itself. So it happens in cycles, and that's what we fight against when we give the drugs or the medicines to treat malaria. So these parasites can identify RBCs from all other cells in the body. And how do they do that? Every cell can be identified because of certain receptors. So suppose, how do you identify your friend's house? Suppose there's a friend who's living in the next block or the next society. And it tells that, okay, come over to my house. How will you identify your friend's house?
By the building.
Some by the building, but let's say all buildings are same. It's a society, every building is same built. It was made by the same architect. So all buildings are same. Then
by asking.
sorry,
By the color.
by the color. Even the color is same.
So by asking someone.
By asking someone. Okay, that's okay. I think putting a address or a name plate is not in fashion these days, I think. So you can just identify someone's house by the address, right? If it says that my house is 136B, We just have to look for 136B, right? And whichever building says 136B or whichever apartment says 136B, you can just knock on the door because you will be sure that this is your friend's house, right? That's called a marker for that address, right? A pin code, a address, or a marker. Do I make sense? Normally,
Yes,
you
sir.
will just call your friend and say that come out, I'm in front of your building, and he'll come down or she'll come down. But it's fine. But when a parasite enters our body, it cannot ask other cells. Okay, so imagine a parasite going to a WBC and asking, okay, where is RBC? I want to attack it. WBCs won't tell, right? So it has to identify RBC on its own. So a parasite, a microorganism, identifies a cell which it attacks by the base on the basis of, so let's say this is the surface of the cell. Cells have a lot of receptors, a lot of proteins that come out of the membrane. Those are called cell surface receptors. So there are receptors on the surface of RBC that these microorganisms can identify and hence attack that cell. When sickle cell anemia happens, the membrane of the RBC changes because of mutation. And you know, the very interesting thing is, if a person has sickle cell anemia, the person cannot have malaria. The chances of malaria drastically goes down. Even if the parasite enters the body, it won't be able to find out an RBC because now the RBC is not exactly what it should be for the parasite to identify. Do you understand that? Is it clear,
No,
people?
sir.
Yes,
Okay.
sir.
Okay, someone said no. So without sickle cell anemia, the shape of RBC is normal. So the surface is normal and the receptors on the surface are also normal. So a parasite can come in and identify a RBC. Okay. But when sickle cell anemia happens, all the RBCs changes to a sickle shape. It has its own problems. It's a disease, but that prevents the malarial parasite to now identify and attack the RBCs. So RBCs cannot be identified. They are no longer identifiable by the parasite Plasmodium that causes malaria. Is that clear, everyone?
Yes.
Yeah. Now, it does not mean that sickle cell anemia is a good disease because it prevents from RBCs. It prevents from malaria. It also has other problems. Okay, so the problem is that you do not get proper oxygen moving around in the body, and you need to get treated for that. So sickle cell anemia is a problem which comes with very low oxygen in the body. But in areas, why I'm telling you all of this story is because in regions like Africa, where malaria is very, very prevalent, it's a big chaos. It's a major cause of deaths of people in Africa. So in African continent, now scientists are seeing that many people are selecting and tolerating this carrier phenotype. So in evolution, half of their RBCs are getting selected. They are selecting a sickle cell phenotype. And these heterozygous genotypes are flourishing more in the population because it provides you almost 50% shielding against malaria. So even if a carrier person gets malaria, the severity of malaria won't be too high, and the person has more chances to survive if the person is a carrier for sickle cell anemia. So evolution of humans, which is still happening, in Africa, it has taken a very interesting route that they've started selecting a heterozygous phenotype for a disease. They prefer to be carriers so that they can fight malaria, which is a big problem in the region. Okay, because malaria, a heterozygous condition shields from malaria. Is that point clear, everyone? Parker? Yep. Iram, Parker, Mariam. Everyone, just note this down. Give me 1 second. And ask me if you have any doubts, please. And this all happens because of a point mutation. Okay, everyone is clear with these two things?
Yes, sir.
Yes, sir.
Let's go to others. What next? Other Mendelian disorders?
Hemophilia.
Hemophilia, perfect. So hemophilia. What does the name suggest you? What is it, first of all? Is it what kind of a Mendelian disorder it is? Autosomal, sex-linked, recessive, dominant, what it is? Come on, people. You have not done the homework. It means. Okay, so it is a sex-linked. This is the first sex-linked disorder. When I say sex-linked, I mean X chromosome linked. Okay, so X-linked disorder, and it is thankfully
Recessive.
recessive. And in this condition, what happens is that you know about blood clotting factors, blood clots in our blood clots. When does it clots? Whenever
During
it
an
comes
injury.
in, yeah, during an injury specifically, because whenever it comes in contact, whenever it comes outside our arteries or veins, it has a tendency to clot, because an injury or whenever it comes in contact with the outside environment, there is a signaling that activates in the blood, which allows these blood clotting factors to activate and make the blood clot through using platelets, fibrinogen, etc. So in that whole signaling pathway, we have lots of proteins that make this signaling happen and finally the blood to clot. Now this clotting also depends on how healthy our signaling system and our all these proteins are. So some people's blood do not clot that fast. Some people's blood clot faster. In certain diseases, the clotting time of blood increases a lot or it does not clots. So those people are more vulnerable to catching infections or deaths due to injuries because they will not be able to clot their wounds. Okay, and clotting is very important for the healing of any kind of a wound or injury, right? So in hemophilia, what happens? One single protein, which is a part of this whole group of proteins that allows blood clotting, is affected and is mutated. And because of this mutation, kids or babies which are born with hemophilia will not be able to clot their blood. Now, this you might seem that, okay, so what's the problem? The problem is we need this blood clotting mechanism every time. Let's say you can protect the kid from injury. So, but very simple injuries, like even if you go to hospital and you are pierced by a needle to give you antibiotic or to give you vaccine or to give you any medicine, if your veins are punctured, blood comes out, right? Then the nurse will give you some cotton and tell you to press that point for some time and then leave, which means your veins clotted by that time. Even if an injection is pierced in these people, the blood will constantly keep coming out because there is no clotting possible. So it's a very, very problematic situation where you're not able to clot the blood. Okay. And people with hemophilia can die of blood loss, even by minor injuries. Okay. So hemophilia is a problem. And it is also known as the royal disorder. Anyone knows why it is known as the royal disorder?
Because it was first found in the royal family.
It was not first found in the royal family. Okay. It is in the royal family. It was running in the royal family for a very long time. That's why it's called the royal disorder. It was discovered much before, but we know that Queen Victoria, if you see the pedigree chart of Queen Victoria, you will find a lot of hemophilic descendants because Queen Victoria was a carrier for the disease. And most of these hemophilic descendants were males because males just have one X. And if a female, so Queen Victoria was, the genotype of Queen Victoria was X affected, X normal for hemophilia. Okay. And because of which, a lot of males, Queen Victoria, that inherited this X chromosome were hemophilic. Okay. Now, it is difficult for a hemophilic person, whether male or female, to live a very longer life or to live beyond the 20s and get married and sire kids. But it's not completely impossible. It is possible with proper care. So, and especially if you are, there is another reason why this disorder could prevail in the royal family for very long and for generations is because if you have so much of wealth, I think you can just take care of hemophilia, right? You just need proper blood transfusion every time and do not allow yourself to get an injury or something. So many of these royal family hemophilic descendants have survived and also had kids, but they could not realize for a very long time that this is a genetic disorder. And if they will keep producing kids, this disorder will keep passing on. Okay. But there is some interesting questions that are asked in entrance from this part, hemophilia. So let me ask you something. So first write down the cause, Hemophilia is caused due to a single protein getting mutated, from the group of proteins responsible for blood clotting. Full stop. Next line. Due to this, the affected individual or the affected person, due to this, the affected person will not be able to clot the blood even after a minor injury. Will not be able to clot the blood even after a minor injury. Okay.
So,
Yes. Yeah.
sir, if a female is carrier, no, not carrier, if she's affected, then it would be very problematic because due to the monthly cycles. Right?
Exactly. But nature has its ways. See, something which is so problematic is not very easy to happen. Now, you raised a very good point, and this is actually an entrance question. So let's prepare some questions for entrance examinations here. So for a female, and I hope others like Others will also participate apart from Iram. This is
So,
Iram's question. Yes, Iram has a question again.
so,
Yes.
but like, do menstrual blood clot?
And do menstrual blood clots? Menstrual blood does clot when it comes out of the body. It should not, and it does not clots when it is inside the body. Yeah. Because menstrual cycle, see, bleeding phase only happens when the endometrium starts breaking down. And when it starts breaking down, the blood vessels which are there also break down. Some tissues also break down. And that is first time when the blood comes out of vessels and comes out of the vaginal tract. And when it comes out, then it clots, of course. Any blood from human body, if it comes out in the open air, it will clot. But till it is inside the body, it won't clot. It should not, right? It's a problem if it clots inside the body. And these kind of clots do form inside the body sometimes due to other problems or disorders. And these blood clots can go and choke any artery or vein, and it can cause a lot of problem. Internal bleeding is a big issue because if you get an internal injury, blood clots there between a artery or a vein and some tissues or an organ. So when the moment it comes out of arteries or veins, some clotting factors get activated and it might clot there. And that clot can go and, block arteries in the brain, in the heart, in liver, kidneys, and cause a lot of problem. Deaths mostly. Yes, but let's come back to Iram's question. So if a female is hemophilic, what should be the genotype? Mariam, you tell. Mariam Khatun. For a female to be hemophilic, what should be the genotype? Come on. It's an X-linked recessive disorder. Hafsa? There's only one way a female can be hemophilic. X affected and X affected, right? Yes, sorry, Parker. I gave the answer before you raised the hand. So you were saying the same thing, right? I assume?
Yes, sir.
Now you tell me, where did this X come from?
The male side.
Yeah, one X must have come from the father, which means the father was hemophilic, right?
Yes, sir.
And other X came from mother, which means the mother was at least a carrier, right?
Yes, sir.
Or herself a hemophilic, which is even rare. So for a female to be hemophilic, the possibilities are very less because you need a hemophilic father to marry a hemophilic or a carrier mother. So the odds are very less for this itself to happen. Do you understand, Iro? It itself is
Yes,
it's
sir.
not very, very prevalent a disease. It's not like every second person in the world is hemophilic. No, it's very rare. So a rare hemophilic man who must have survived till the age of getting married had such a bad luck that he ended up married either a carrier hemophilic female or a hemophilic female. So odds are very, very less. That's why you will not find hemophilic females, too much hemophilic females in the world. Okay. Do you understand?
So if it is there, then what would happen?
Yes, then it will be very difficult for that female to reach puberty. Females will die as kids. And even if you say like all the luck worked for that particular individual and let's say a lot of money and everything worked and somehow she managed to reach puberty and menstrual cycle began, then it's going to be even huge a chaos and body won't be able to tolerate. First, she's hemophilic. Second, she's menstruating. So menstrual flow will be huge and whatever damage will happen in the uterus won't be able to heal back properly for the next hemophilic cycle, or sorry, for the next menstrual cycle and so on. So it's a total mess. Do you understand? So hemophilic kids die very early, normally. But this question comes in boards. Write down the question. Why the possibility of a hemophilic female? Why the possibility of a hemophilic female being born is extremely rare? Why the possibility of a hemophilic female being born, hemophilic female child, you can write, being born, is extremely rare? Hemophilic male childs are more prevalent, okay? Because all you need to make a hemophilic male child is a carrier mother. Yes or no? If a mother is carrier, the mother won't know because she will not show the symptoms for the disease. That's why these days, since all these recessive diseases traits are getting accumulated faster because of cultural practices, and many cultures also encourage consanguineous marriages, which is marriages among close or close cousins or relatives. So over generations, the chances of recessive genes getting accumulated in the broader family is more. So now what happens is a female will never know if she is a carrier for hemophilia, okay, unless and until she gets her herself tested for that gene. That's why governments across the world and medical team, scientists, doctors across the world are trying to create awareness that if you are going to have a baby, please go for a genetic test through amniocentesis of the baby. They will not reveal the gender, but they will check for symptoms. They will check for chances of hemophilia. Even before planning a baby, the mother, the women, and the man, they both can go and get themselves tested for carrying these alleles or not. So if a mother can get to know that I have chances of, I'm a carrier for the hemophilic gene, then they can be more cautious during the pregnancy. And if it is a girl child, she don't have to worry much. But if it's a boy child, then it will be good if she knows whether the kid is having hemophilia or not. Because it's better to know it in the first week of pregnancy, and you can medically terminate the pregnancy, because it's always better to bring a healthy child to the world than a kid who's going to suffer. You know for sure that he's going to take birth and going to suffer a lot and die very early, right? So medical science helps that thing. Okay. Also, around the globe, cultures, because of as more and more people now believe in science and function according to scientific things, even scientific medicines and habits and behaviors. People are fitness freak. People understand the reasons for many lifestyle related diseases as well. So obesity, diabetes, and in the coming times, the problems of the heart, liver, and kidneys, they are not just age related anymore. So I urge all of you to just create this awareness for yourself, for your families, to incorporate healthy habits, eat healthy, live healthy, because you know what's going to happen if something gets accumulated in your body, because we have evidences for that. That's what you are learning here, right? That's what biology tells you about. So biology is the science of what's happening inside you. You are basically studying your own system on in chapters, on pages here, right? So you all wrote the question, why is a female hemophilic child extremely rare?
So,
Yes.
Yes, yes, yes,
so I'm so sorry,
So-
I still have a question.
yes, of course. Questions are,
So
see,
one
this is how you learn the most. So all questions are allowed. Just to finish the syllabus, we don't need a year, believe me. because the same syllabus is also covered in some crash course time where you just go for 2 months back forth and you just finish the syllabus. We need 1 year to understand these subjects. Why do you think your school teaches all of this in 1 year? This is not a year worth a year syllabus. This is what at max 5 months, 4 months. But to understand it takes time. So that's what we do in the class. Sometimes I also get delayed in syllabus, but that's not a problem. It's fine. So do ask questions. Do not hesitate in asking questions, people. Yes, Iram, go ahead.
For a affected hemophilic female, there's a menstrual loss, like if she hits puberty, right? But in menstruation, there's the inner lining, which is the endometrium breaking down, right? So if that breakdowns and comes during the menstruation, so where does the excess blood come from?
Where does the excess blood come from? The blood.
Because she continuously bleeds like the female.
Yeah, it's a good question. So for that, let's say this is the uterus lining. Okay, this is the myometrium and the perimetrium. And what I'm going to draw with red is the endometrium. So this is the endometrium, right? That becomes. So from the myometrium, blood vessels come inside and make this endometrium more and more thicker. You understand?
Yes, sir.
Now, when menstruation happens, this whole endometrium kinds of sheds out, right? Breaks down and comes out along with tissues, cells, and blood vessels, right? But the blood vessels that came from the myometrium into the endometrium, they needs to clot, right? Faster within a day or two, so that within 3-4 days, blood flow will stop and you will start rebuilding again. Do you understand that?
Yes, sir.
But these blood vessels that got ruptured, they won't clot. That's the problem. Any kind of injury. So menstruation, if you think biologically, it's an injury only, right? If I get my skin bruised, a layer of my skin will come down, right? It's an external injury. But blood will clot, tissues will again heal up, and I will make a new skin. Make sense?
Yeah.
Think of menstrual cycle also as an internal injury happening every month. So the female needs to lose blood, clot, make up things, again lose blood, break down, wear and tear, then clotting. It has to happen. So without clotting, menstrual cycle, you cannot imagine. It's a very painful and difficult thing. And female hemophilic kids do not live that long, I think. I don't know if if they're carriers, then they are normal only. So female carriers do live. And Queen Victoria was one such female carrier. But getting a female hemophilic child up to an age of menstruating is, I don't think it's possible. Yeah.
Yeah. Okay.
Does that answer your question? Does it make sense?
Yes, sir.
Okay, perfect. Good. So this question is done. And this question can also come in entrances asking you different versions of why hemophilic female is not possible and giving you reasons in statement. You have to choose the right reason. So the right reason is you need at least a hemophilic father and at least a carrier mother for a hemophilic female child to take birth, which is very rare. Okay. And it is also rare because these fathers won't live up to the age. A carrier mother can live, but hemophilic fathers, it's not, it's unviable for them to live up to the age and getting married and giving rise to kids. Okay, cool. So hemophilia is clear, everyone? Any doubts in hemophilia?
Yes, sir.
So this was a sex-linked recessive. There is another sex-linked recessive disorder for which the situation is very different. We see both male and females, which is called color blindness. So I'm giving you now today 2 things for your homework: color blindness and thalassemia. Thalassemia is autosomal recessive. Color blindness is sex-linked recessive. I want you to read these 2 diseases and try to make their pedigree if you can on your own. Try to come up with any pedigree, take any male and female, and try to make a pedigree for thalassemia and color blindness. And then we have to finish chromosomal disorder in the next class, okay? Because I think we are out of time. But I'll give you some context if you all can give me 2 minutes, which will help you when you are reading on your own. So your homework is color blindness. Color blindness is where people can't differentiate between colors. Mostly color blindness is of red and green color. Sorry, red and blue color. Also green color sometimes. That's color blindness. Yeah, red and green. Blue color blindness is rare, but it does happen. And thalassemia. Okay, so these two you have to read because this is sex-linked and recessive. This is autosomal and recessive. You might have noticed that we are not studying dominant, whether sex-linked or because dominant diseases are very difficult to study because people die very early if there is a genetic dominant disease. After this, the last part of this chapter that we will try to finish in the next class is chromosomal abnormalities. Now, chromosomal disorders are not called Mendelian because they do not follow the Mendelian rule. Here, it's not a gene that is problematic. In fact, no gene sometimes is mutated. The only problem is that you have a new copy of the whole chromosome. So the whole chromosome is either excess or deficient. So if any single chromosome is in excess, you call it aneuploidy. So two things, try to read these two terms. Aneuploidy, it simply means a gain or loss of a chromosome, of one chromosome, okay? It can be any chromosome. A gain means one extra copy, a loss means just single copy present, okay? That's aneuploidy. Second thing is polyploidy. Polyploidy is not tolerated in humans at all. There cannot be a polyploid human. Polyploidy means whole set of chromosome doubled or extra, which means instead of 46 chromosome, 23 pairs, you will now have 46 pairs, which is 92 chromosomes. So that's not possible in humans. Even if an embryo has that embryo will die right there. It will not grow into a fetus. Okay. Polyploidy can only be tolerated by plants. So you can write down it is tolerated by plants. And some animals are exceptions, but not humans. Okay. So mostly focus on aneuploidy for the next class. And in aneuploidy, you will see some disorders that happen due to aneuploidy are Down syndrome, Klinefelter, Turner, and Tau. 4 syndromes we will study in this, in aneuploidy. Down syndrome, Tau syndrome, Klinefelter, and Turner syndrome. Okay. So I believe you all will read this and come back in the next class. Yes, no?
Yes, sir.
Yes. Hafsa, can you just stay for 1 minute and others can log out?
Thank you, sir. Bye.
Yes.
Yeah. Bye, bye
Yes,
everyone.
sir.
Yes. How are you doing, Hafsa? How do you like the class? Like, do you understand? Were you able to catch up?
Yes, sir.
Yeah. Okay, cool. So since you have joined a little late in the schedule, I just wanted to know what is the status of your school syllabus so that I can, readjust or reorient according to that. And we also have to figure out if you at what time some extra classes can be scheduled for you. So what is the status of your school, Hafsa? Which chapter are you
Sir,
doing?
we are in chapter 8, human health and disease. We have done up
Okay.
to cancer treatment.
Okay. And from chapter 2nd to chapter 7th, you have done all the chapters?
Yes, sir.
Okay. And from 9th to the rest, 16th, no chapter has been started, right? So it's going
No,
in
sir.
one series. Okay, perfect. That makes life a little easier. So you have already done in your school chapter 5, 6, and 7, right? Which is
Yes.
genetics and evolution. Okay, great. So how about, and when is your next exam? And what is that? Mid-sem, half yearly? What is that?
So it will be in the starting of our school, that is 27. It's like, I guess it's first
July,
term. Yeah. No,
first
sir,
term. And what
August.
is this? August, 27th August.
Yes.
Okay, so we have time. And what is the syllabus? Any idea? What are they going to ask you?
It's going to be like the whole syllabus, whatever we study up to the,
Whatever. Okay.
yeah.
So
Yeah.
okay, so clearly this will also be a part of it. Okay, cool. So then there's not much of problem. So we can continue with this. And for chapter 2nd, 3rd, 4th, and much of 5th, we will see for some extra classes that we can, we have to give some extra classes to you and some other people who joined a little late. So we will try to keep them half an hour before the actual class or half an hour after the actual class in the coming times. Okay. So we'll try to cover what you missed. Okay, Hafsa?
Okay. Yes,
Any
sir.
feedbacks for me? Uh,
Yes, sir.
was the
Um,
class understandable?
yes, sir.
Yes. Yeah. You have any feedbacks for me? Anything I should
Yes,
change
sir.
or should include? Yeah, tell me.
Yeah, I was able to understand. And
Mhm.
so I wanted to ask, if you had a previous student, like last year's student named Hishma,
Last year students? What? Sorry?
named Hishma. Uh, like I wanted to ask if she was your student, Fatima Hishma.
Fatima.
Yes, sir.
Yes, Fatima was my student. Yes. How do you know Fatima?
She was the one actually who recommended me to join this.
Oh, that's great. So how
Yeah,
do you know her?
yeah,
Like, is she your friend? She's a
yeah.
relative,
She's my
neighbor?
friend.
Ah, she's a
Yeah,
friend. So you're both
yes,
from same school.
yes, sir. She's
Oh,
my
that's
senior.
great. Oh, great. Yes. Say my hi to Fatima and tell her that I
Yes,
do
sir.
remember
Sure.
her and hope she's doing good. Okay. Just convey my
Yes, sir.
regards. And great. You're coming through Fatima. Then I think it's